For centuries, we've understood that kids get traits from their parents. That's obvious. But the how, the actual physical blueprint for life, well, that remained one of the biggest mysteries out there. So let's dive into this incredible scientific detective story and see how we finally cracked the molecular basis of inheritance.
Really, it all comes down to this one question. What is the secret code that connects every single living thing on this planet? Think about it. Whatever this code is, it has to be able to store a mind-boggling amount of information, copy itself almost perfectly, and somehow guide the creation of a living, breathing being.
For decades, scientists had a prime suspect, but they were wrong. Most of them put their money on proteins, just because they seemed so complex. But the real culprit was hiding in plain sight. The hunt was officially on for what they called the transforming principle, some kind of molecular ghost that could pass traits from one organism to another.
And what's so cool is how this case was built, piece by piece. It starts back in 1928, when Frederick Griffith finds this first major clue. Something from dead, nasty bacteria could actually transform harmless bacteria into killers. Fast forward to 1944, and Avery, McLeod, and McCarty collect more evidence, pointing the finger squarely at one molecule, DNA. But the scientific community wasn't buying it. DNA just seemed way too simple. It was going to take one final brilliant experiment in 1952 to slam the case shut.
Okay, this is the experiment that totally sealed the deal. Alfred Hershey and Martha Chase used viruses, these little things that infect bacteria. Now, to figure out what the virus was injecting, they used this incredibly clever labeling trick. On the left here, you see they tag the viral proteins with radioactive sulfur. That's the red capsule. Then, on the right, they tag the viral DNA with radioactive phosphorus, which you see in blue, because phosphorus is in DNA but not protein. So after the viruses did their thing, they separated them from the bacteria, and the result? It was completely undeniable. The radioactive phosphorus, the DNA, was found inside the bacteria, already reprogramming them. And the radioactive sulfur, the protein, was just left outside, clinging to the surface. Boom, that elegantly proved that DNA is the molecule carrying the instructions. Case closed.
So, knowing DNA was the stuff of genes was a huge breakthrough, right? But it just opened up a whole new mystery. How does it actually work? How can this one molecule store and copy so much information? Well, it turns out the answer was hidden in its shape.
So let's break down the evidence. DNA is basically a long chain, and it's made of these repeating units called nucleotides. Each one has 3 parts. You've got a phosphate group, a sugar called deoxyribose, and then one of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine, or, as we all know them, A, G, C, and T. And this is the answer. In 1953, using some absolutely crucial X-ray data from the scientist Rosalind Franklin, James Watson and Francis Crick proposed this iconic double helix structure. And it was a complete and total game changer. All of a sudden, everything we knew about genetic material started to make perfect sense. Now, the structure has a few key rules. The 2 strands run in opposite directions, kind of like a divided highway, and a sugar-phosphate backbone gives it its strength. But the real magic, the secret sauce, is on the inside. The bases pair up in this very specific way. A always pairs with T, and G always pairs with C. This base pairing rule, well, it's the secret to pretty much everything. Okay, so once the double helix was figured out, the next piece of the puzzle just fell right into place. How does the cell actually read this code and use it to build stuff? The structure itself pretty much screamed the answer. Francis Crick laid out what he called the central dogma. It's this simple but really profound idea that information flows in just one direction. It goes from DNA, gets transcribed into a messenger molecule called RNA, and then that RNA gets translated into a protein. Think of it like this. DNA is the master blueprint in the head office, RNA is the working copy you take to the job site, and proteins are the actual machines and workers that do all the work. So let's get this straight, because there are 2 key processes here that sound alike but are totally different. Replication, that's when the cell needs to copy its entire library of DNA, like before it divides. Transcription, on the other hand, is like just checking out a single book from that library. The cell copies one specific gene into a temporary message, an mRNA molecule. Let's take a closer look at replication. An enzyme comes in and literally unwinds the double helix, creating this replication fork you see here. And thanks to those super strict base pairing rules we talked about, each of the original strands serves as a perfect template for building a brand new partner. We call this a semi-conservative process.
because each new DNA molecule has one old strand and one new one. It's an incredibly elegant and super accurate way to copy the code.
So all these foundational discoveries, figuring out DNA was the stuff, solving its structure, understanding the central dogma, they unleashed a total revolution. We went from just hunting for the code to actually being able to read it, and that brought us into the modern era of genomics.
And the scale of our own code is it's mind-blowing. The Human Genome Project showed us that our instruction book has over 3.1 billion letters. That's an entire library of information packed inside nearly every single one of our cells.
But get this is one of the most surprising things they found. Despite all the incredible diversity we see in people all over the world, 99.9% of those 3 billion letters are exactly the same in all of us.
And maybe the biggest shock of all? Less than 2% of our entire genome actually contains the instructions for making proteins. For years, scientists called the rest of it junk DNA. Turns out, it's not junk at all. We're now realizing it plays all these crucial roles in controlling how and when our genes get used.
And this is so cool because it brings our whole detective story full circle. The little differences in that non-coding, repetitive DNA give each of us a unique genetic profile. And that, right there, is the basis of DNA fingerprinting. You can see it on this slide. The pattern of bands from the crime scene, marked C, is a perfect match for suspect B. Our ability to read the code of life now helps us solve modern crimes. So, from a mysterious, transforming principle to the double helix, and now the entire human genome, we have come an absolutely incredible way. We've learned to read the book of life. And that leaves us with a really profound final question, especially as new technologies are coming out that let us not just read but actually edit this code. What should we write next?